Hello, everyone. Welcome to today's live broadcast of multiple IHC and novel monoclonal antibodies as enabling tools for the study of tumor immunology. I am Sam of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots, the leading social media site for science professionals, and sponsored by Cell Signaling Technology. Founded by research scientists in 1999, Cell Signaling Technology is a private, family-owned company headquartered in Danvers, Massachusetts, with over 400 employees worldwide. Active in the field of applied systems biology research, particularly as it relates to cancer, CST understands the importance of using antibodies with high levels of specificity and lot-to-lot consistency. It's why they produce all of their antibodies in-house and perform painstaking validations for multiple applications. And the same CST scientists who produce antibodies also provide technical support for customers, helping them design experiments, troubleshoot, and achieve reliable results. See more at www.cellsignal.com. Before we start, there are a few instructions. We want to hear from you during this live broadcast event, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right, or use the Q&A button. We'll make sure to try to resolve any issues. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Now let's get to today's presenter. We're proud to welcome Dr. Matthew Silver. Dr. Matthew Silver leads the Translational Assays Group at Cell Signaling Technology, where he focuses on the development of both the translational research reagents and multiplex assays. Matthew received his PhD in biochemistry from the University of New Hampshire, where he studied neuroendocrine regulation of reproduction. His subsequent postdoctoral studies were performed at Wyeth Research within the Department of Inflammation. I'll now turn it over to Dr. Matthew Silver.
Thank you very much for the introduction. Again, my name is Matt Silver, and my presentation today is going to be focused on the use of multiplex IHC and novel monoclonal antibodies to study tumor immunology. So my presentation is going to be broken down into three sections. I'm going to start with an introduction into multiplex IHC and how we're performing multiplex IHC. I'll then move into IHC antibodies that we've been developing that are relevant for immunotherapy targets. And then I'll finish up with some information on panel development. So starting with multiplex IHC, the rationale for multiplex IHC is straightforward. It comes down to the simple fact that there are a lot of biomarkers and there tend to be a limited amount of biopsy materials. The benefits for multiplex IHC really come down to the fact that tissue samples are small and typically rare, for example, with fine needle aspirates. You can ultimately acquire more information per slide and per section using a multiplex IHC approach. And while not necessarily as good as what you get with a traditional chromogenic IHC, it's important to note that you certainly maintain much better tissue context than what you get with other grind and bind multiplex assays such as next-gen sequencing, PCR, or mass spec. So there's a lot of benefits to multiplexing. Of course, there are a number of challenges as well. For example, fluorochrome spectral overlap can be an issue that can limit the degree to which you can multiplex. Tissue autofluorescence, which can come from a number of things, including the fixation process itself, can wreak havoc with the sensitivity of your assay. You need good antibodies that work well in formalin-fixed, paraffin-embedded tissues. That's really key to the whole process. But more than that, you need good antibodies that work well together in a multiplex setting. And then down the road, pathologist acceptance will be an issue as multiplex IHC makes a transition at some point from a research, translational research mode into a potential clinical assay. So at CST, we've been working to make multiplex IHC routine, which we feel comes down to the combination of your imaging platform, the detection reagents that you're using, and of course, best-in-class antibodies. And specifically in which the ways these different areas address some of the challenges I mentioned on the previous slide. The imaging platform that you choose can help address fluorochrome spectral overlap and tissue autofluorescence. Detection reagents help with multiplexing with signal amplification to enable your assay through higher sensitivity. And then best-in-class antibodies speak for themselves, where you really need highly validated quality antibodies that work well in formalin-fixed paraffin-embedded tissues. So the solutions that we've been working with on the imaging side, we have primarily been working with multispectral imaging as a platform, which enables us to multiplex with 5+ colors, and it also helps us eliminate autofluorescence that we get from the tissue. You do not need to use multispectral imaging if you're doing a 3-color assay, but we tend to try to push the colors up again to 5+, in which case the multispectral imaging is a key enabling technology. The antibodies, again, speak for themselves. You need the good antibodies that work in formalin-fixed paraffin-embedded tissues, which would be CST antibodies. And then on the detection reagent side, we work with a couple of different approaches. We're using a Tyramide-based multiplex approach, which is a multiplexing approach that provides a tremendous amount of signal amplification, and it does this in a way that is antibody, species, and isotype independent. And we also work with some modified monoclonal antibodies that enable multiplexing using a secondary-based detection system. So for the presentation that I'm going to do today, I'm going to focus in on the detection reagents and primarily talk about our use of Tyramide. So on the detection reagent side, it's really the key to multiplexing with signal amplification. And so when you're talking about doing a 4-color assay, which to be clear would be 3 targets plus DAPI, it's pretty straightforward. For example, if you are going to look at a HER family member panel, so looking at EGFR, HER2, and HER3 as an example, it's pretty easy to find a rabbit monoclonal and a mouse IgG1 and a mouse IgG2A to enable your multiplexing. However, you tend to run into trouble when you move into a 5+ color assay. And you can take a couple of different approaches. You could look for alternative isotypes, which in some cases will work out. However, there's really a limited number of high-quality rats or mouse IgG3 or chicken antibodies. And so if you're looking to develop a broad series of panels, this can become a hurdle that can be difficult to get over. Alternatively, you can use direct conjugates, in which case you're only limited by the number of fluorochromes that you have access to. However, when working in tissue, directly conjugated antibodies tend to lack the sensitivity required to really develop a robust panel. And just as an example, the data shown here on the right are looking at our EGFR staining using our rabbit monoclonal with a secondary antibody, so your standard primary-secondary based assay shown right here. And the conjugated version of the antibody is shown here on the left. And this is looking at A431 cells shown in red versus MCF7 in blue. And you can see while you certainly can detect signal with the conjugated antibody, you take a dramatic hit in sensitivity here measured by mean fluorescence intensity per cell. So in some cases this might work out just fine and you'll be able to detect your target. However, in many, the sensitivity loss will wreak havoc. So the solutions that we've been working with, again, are the use of Tyramide signal amplification and modified monoclonal antibodies. On the Tyramide, again, is a serial staining approach that leads to a high degree of signal amplification, whereas the modified monoclonals is a parallel staining, which a more traditional parallel staining, which enables a moderate degree of secondary-based antibody signal amplification. And to give everyone a sense of the difference in signal intensity that you'd expect with these different approaches, on the left here we have what you'd see with a conjugated antibody, where you have your epitope and your antibody that is conjugated to a certain number of fluorochromes, which typically would be 3 or 4, and that provides the signal that you would detect. You get amplification when you're using secondary antibodies now because you have multiple conjugated secondary antibodies that are binding your primary, so you increase the number of fluorochromes per epitope. When moving to the Tyramide signal amplification approach, on the other hand, you have a very dramatic degree of signal amplification because this detection mechanism is based on an enzymatic reaction. Tyramide itself is a small phenol, it's a basic molecule that you can conjugate to a number of different things, including haptens or fluorochromes. And then in a reaction that's catalyzed by HRP, you create a highly reactive free radical form of tyramide, which will create covalent bonds with electron-rich regions in close proximity to the reaction, which typically would be a tyrosine residues on the antibodies, on and around the epitopes. And so again, this enzymatic approach leads to dramatic amplification of your signal, which you need to control very carefully, but if handled properly can be a very powerful technique. And here's an example looking at our ALK antibody D5F3, staining H3122 cells, which express the EML4 ALK translocation, and T47D cells, which are ALK negative, using our standard IF approach of using a primary and secondary antibody up top here, versus the Tyramide protocol on the bottom. And you can see with the standard approach, you can detect the signal, although it's fairly weak, and it's clean in the T47D cells. But when you move to the Tyramide-based approach, you can see a very dramatic increase in signal intensity while maintaining the negative signal in T47D cells. On the right, we have the quantification of these signals, looking at mean fluorescence intensity per cell, where H3122 cells are in red and T47D cells are in blue. And you can see this very dramatic increase in signal intensity in the H3122 cells with a very minor increase here in the T47D cells. So two other important things to point out in this slide. In addition to the drastic increase in signal-to-noise and signal intensity, the actual imaging times are much faster with the Tyramide protocol, where we have a 10 millisecond exposure compared to 75 milliseconds for the standard protocol. And then we're also using less antibody because of the strong amplification with the detection reagent. So we're using the antibody at 1 to 1,000 with Tyramide compared to 1 to 250 with the standard imaging approach. So this is Tyramide in cell pellets. We've validated this approach in tissues as well. And so this next slide just highlights a number of tissues that we've stained using the Tyramide approach, simply to illustrate the point that this isn't just about staining cells, and it's very much compatible on a tissue, formalin-fixed paraffin-embedded tissues as well. So that's how TSA works in the singleplex setting, and it's perfectly suited for multiplexing as well through the introduction of an additional microwaving step, or some step that ultimately will disrupt the antibody interaction with your sample. We tend to use microwaving because it fits into the workflow very nicely by repeating antigen retrieval, but alternative heating methods and acid stripping are alternative approaches to doing this as well. So the way this would work is you have your epitope with your primary and secondary antibody, and you go through your detection reaction, activating Tyramide, which becomes deposited around your sample. You then introduce a microwaving step, which would wash away your primary and secondary antibody, and your covalently bound Tyramide fluorochrome conjugate remains behind on your sample. You then simply come in with your next antibody, and the species and isotype no longer matters because your primary and secondary from the previous step have been washed away. And now you include a Tyramide conjugate to an alternative fluorochrome. So if initially it was Alexa 488 as an example, you can now come in with Alexa 647 and go through your reaction, another microwaving step, and simply repeat until you use the number of fluorochromes that you can handle on the imaging side or the number of fluorochromes that you need for the particular experiment or panel that you are currently working on. So just a couple of comments about the Tyramide signal amplification multiplex protocol. Again, it's a serial stain, so you have your initial microwave treatment, which is your antigen retrieval step. You go through your first stain and repeat microwaving and your second stain in this serial fashion. The actual primary and secondary antibodies and Tyramide reactions live within each staining step, and it is ultimately a fairly long procedure where it's about 3 hours per color that you're working on. And obviously that can translate to a very long day if you're looking at a 3 or 4 plus color assay, but you can introduce an overnight break in between by incubating your slides and blocking buffer without consequence on your signal intensity. And so one thing that I think is very much needed is the development of automated protocols and solutions for this approach, because again, it is a very long protocol. A couple comments on optimization. One of the really important steps as you're going through and developing panels is looking at the position within the staining series. So on one hand, your fluorochrome is covalently bound to your sample, but as you go through the multiple microwaving steps, you can lose, sort of chip away some of your conjugated fluorochrome, and so you can experience some signal loss as you go through the microwaving steps. And so you might want to put your weaker antibodies or weaker signals later in the staining series. However, you need to balance that with the fact that as you go through multiple rounds of microwaving, as you might imagine, you can experience epitope damage. And so it's really important as you go through and develop these panels that you look at each antibody and each position across the staining series to control for potential signal loss and epitope damage. So as an example, here looking at ROS1 staining in a 3-cell pellet mix that we developed, which includes around 1/3 of the cells being HCC78 cells, which express ROS1. When we look at ROS1 staining in the first position, you can see roughly 1/3 of the cells staining, so an example pointed out right there. However, when we look at ROS1 in the fourth position, now all we have is this one cell left, and it took quite a bit of scanning to find the cell, so we completely lost the signal after the third microwaving step. So it's really important to go through and get an understanding of how your antibodies are going to perform in each step along the staining sequence. So at this point, we have a quick poll question that we're going to put up. If you could kindly answer this poll question, we'd greatly appreciate it. Okay, so with this in place, with your imaging platform and detection reagents and best-in-class antibodies, it's now time to develop panels when you have all of these things in place. And there are a nearly limitless number of panels that are of interest. A few of many panel concepts are illustrated here that we've been working on in immunotherapy, looking at ALK, ROS, and MET, or EGFR mutations in lung cancer, ER, PR, and HER2 in breast cancer, and her family member panels. And we're interested in signaling node panels and many others, including tumor resistance, looking at ERCC1, RRM1, and TS. For today's presentation, I'm going to focus in on immunotherapy, looking at a number of targets where you ultimately can address questions around tumor infiltration, checkpoint control co-expression, and basic immune cell characterization. So just in way of a very brief introduction, I think as everyone's aware, cancer immunotherapy is an explosive field, and it's all about empowering your immune system to fight cancer. And the concept here is really focused on empowering, again, your immune system to fight cancer. And for a long time, the field had struggled with the question of why doesn't your immune system take care of the problems, the tumor itself. And obviously, part of the problem is that tumors are self-derived. However, there are a lot of changes as tumors evolve, which lead to a great number of neoantigens that the immune system should be able to home in on. And so one way to think about it is the concept called immunoediting, which consists of 3 phases: elimination, equilibrium, and escape. So in the beginning, the immune system will challenge the tumor in order to eradicate it. However, when this isn't perfectly successful, the tumor and the immune system find themselves in a balance, which is the equilibrium phase, where the tumor is under constant pressure from the immune system, which drives selection of immune-resistant clones. And in time, these take over, and this leads to the escape phase, where tumors can propagate even in the context of an intact immune system. And so after years of research, several mechanisms have been identified that will assist the immune system. This includes manipulation of immune checkpoint proteins, amongst other approaches like vaccination and T-cell engineering. But today I'm going to focus in on the immune checkpoints, which typically help maintain self-tolerance and protect tissues during immune response. However, it's the dysregulation of these checkpoint proteins in tumors that can lead to immune resistance. And so there are a number of these proteins, many of which are depicted on this slide, where we have a T-cell depicted on the bottom and an antigen-presenting cell depicted up top. The T-cell receptor is shown right here in the middle of the slide, which, when stimulated, will activate T-cells, which is only done in the context of CD28 co-stimulation. And everything else on the schematic plays a role in fine-tuning this response, either in a positive or a negative way. And so these are all immune checkpoint proteins, and essentially all of these are being investigated as possible drug targets, where you'd either block an off signal or stimulate an on signal. And so, of course, there's an enormous amount of investment and effort currently going on into the development of therapies for these targets. However, we recognized a few years back that there is a large gap in the field, which is that there's a lack of high-quality, commercially available IHC antibodies for these cancer immunotherapy targets. And so we developed the objective of developing a comprehensive pipeline, primarily of rabbit monoclonal antibodies, where all of these projects and efforts would be IHC-driven. So again, these targets aren't new. They've been around for a long time, and there are a lot of great flow antibodies, but IHC has been a gap. And then a key to all of the development work that's been going on here is collaboration. So working with key labs in academia and pharma as we develop these IHC antibodies. We initially had to prioritize because there are a lot of potential targets to go after, and so we started with PD-L1, where again the development objectives were IHC, and we're looking for human reactivity due to the lack of commercially available PD-L1 IHC antibodies a couple of years ago. And so this webinar is not primarily focusing on antibody validation, but just to give everyone a sense of how we go about developing antibodies. We're often using Western blots for initial screens using positive and negative lysates before moving into IHC on similar positive and negative formalin-fixed paraffin-embedded cell pellets. And then we ultimately move into IHC testing using control and cancer tissues. So starting with the Western, here's some example Western data for looking at our PD-L1 antibody E1L3N, where we are testing lysates from Carpus 299, SUP-M2, and PC3s. Cross-reactivity testing is a really critical component to the validation work. So here we have a series of transfection cell lines, where we have cross 7 cells that were mock transfected or transfected with human PD-L1, PD-L2, PD-L3, or PD-L4. And you can see that our clone E1L3N only detects PD-L1 and is nice and clean in the lanes containing the other family members. And then we have a MIC tag here on the right showing the expression of all of the different constructs. We then move to IHC testing, where here we have Carpus 299 cells and PC3 cells that were formalin-fixed and paraffin-embedded. And so we see a nice staining differential between these two cell lines, where Carpus 299 cells express PD-L1, but untreated PC3 cells do not. And from here we move into human placenta, which is a nice control for PD-L1, where you'd expect to see staining in the syncytiotrophoblask layer of the placenta, which is the outer section, which is staining here in brown, but it should be negative in the stroma and villi and the internal parts of the structure, which is nice and clean, as you can see here. And from there, we moved into screening a number of normal and cancer sections. Here is an example looking at human lung cancer that's staining very nicely with our antibody. And then further validation comes post-release, where the antibodies are used by a number of investigators. And here's an example from a recent publication that was demonstrating that our clone E1L3N combined PD-L1 in the tumor, as well as immune cells in the stroma, as well as alveolar macrophages shown in panel C, which is an important point to show that the antibody combined PD-L1 in a number of different cell contexts. So in addition to the initial clone that we released, we have a number of different PD-L1 antibodies that we have been working on and have recently released. We have our PD-L1 clone E1J2J, which binds PD-L1 in the extracellular domain, and some example staining is shown here on the top two tissue sections here of human placenta and human colon cancer. And we've also recently released a mouse monoclonal antibody, clone 405.9A11, which, like E1L3N, binds PD-L1 in the intracellular domain. And so a similar set of example data here on human placenta and human lung cancer. And to round out the PD-1, PD-L1 family, we've also recently released a PD-1 IHC antibody, the mouse monoclonal antibody clone EH33, and some example staining shown here in human tonsil, And we've also released our PDL2 antibody clone D7U8C, and again, some example IHC staining, this time looking at Hodgkin's lymphoma. And of course, we're very interested in targets outside of the PD1, PDL1 family themselves. And so we have a number of antibodies that we have validated for IHC and have released over the last several months, including B7H4 and B7H3 antibodies, and a new IHC antibody for VISTA, which is an interesting new immune checkpoint control protein that's related to both B7 and CD28 family members, and TIM3 was recently released as well. And we're looking into alternative spaces such as immunometabolism as well, with our IDO and CD73 antibodies, or CD73 is important in adenosine metabolism. And then, of course, it's really important to have good coverage of immune cell markers with a series of antibodies that we've released and that are coming out, looking at your standard markers such as CD3 and FOXP3 and many others as well. And so the point is, with a broad portfolio in place, it's now possible to start working on panels within the immuno-oncology space. And so, as an example, I'm going to illustrate how we have developed an immune checkpoint control panel. In this particular case, we're going to be focusing in on PD-L1, B7H4, and VISTA, all of which are important immune checkpoint control proteins. And we're looking at these in the context of CD8, which is a marker of effector T cells, and cytokeratin as well, which is an epithelial cell marker. And so for the next series of slides, I'm going to run down our optimization, and so how we set up the panels themselves. And then I'll show some data on screening of a small cohort of breast and ovarian cancer patient samples that we have performed. So as I mentioned a couple of minutes ago, there are a number of important steps along the way as you validate panels, which are illustrated on the top of the slide here. And it all begins with IHC antibody development. So developing antibodies or acquiring antibodies that work well and that are validated for use in formalin-fixed paraffin-embedded tissues. From there, we move into experiments titrating the antibodies using the TSA-based detection method. This leads into order optimization experiments. So again, where in the staining series does each antibody sit? Ultimately, this then leads to antibody fluorochrome pairing before moving into control cell pellet testing and then ultimately tissue testing. So here we have on the left our PD-L1 antibody titration. So again, looking at carcass 299 cells and PC3 cells using our clone E1L3N, where we have mean fluorescence intensity per cell on the left and the dilution point across the x-axis. And so we ultimately walk down the titration series to find the optimal balance between signal in your carcass 299 and PC3s, which in our hands was 1 to 1,400. Next, we moved into order optimization, again looking at our PD-L1 clone, where you can see mean fluorescence intensity per cell, again on the y-axis on the left, and a signal to noise on the right. And across the x-axis, we have the position within the series, where we have the first all the way to the fifth position. And so here you can see there's a dramatic impact of the position on signal intensity and signal to noise as well, where we're actually seeing an increase in signal to noise as we go deeper into the staining series. From here we move into the antibody fluorochrome comparison, where again we have mean fluorescence intensity per cell on the y-axis, and across the x-axis we have the different fluorochromes that we have been using for these experiments, which include Alexa 350, FITC, Alexa 594, Psi3, and Psi5. And you can see there's a fairly dramatic impact on signal intensity based on the fluorochrome that you're using. So this is actually a really important step where you can have a big impact on balancing the signals within your panel. As you can imagine, this is a nice opportunity to pair your stronger antibodies with weaker fluorochromes and your weaker antibodies or weaker signals with stronger fluorochromes in an effort to balance out the signal intensities within the panels themselves. So that was a quick rundown using PD-L1 as an example. And so this table here shows the panel itself, where we have VISTA in the first channel with Psi3, which will be shown in yellow in all the subsequent images. In the second staining point, we have CD8 with Psi5, which will be shown in red. Then B7H4 with Alexa 594, which will be shown in magenta. And this will be followed by PDL1, which we coupled with FITC, which will be shown in green. And then cytokeratin is in the fifth spot with Alexa 350, and this is going to be shown in cyan. And so we ultimately chose PDL1 in FITC, despite the fact that it wasn't the strongest signal, as you'll see here on the left. And this was done ultimately because of the need to balance the panel. And so we didn't need PDL1 in the strongest possible channel. We reserved that for the VISTA clone. So an example staining in a control tissue is shown here. This is human tonsil, which we'd expect to see staining of all of these markers, where you can see PDL1 expression in green, which overlaps in part with cytokeratin staining, which is here in cyan. And you can see VISTA-positive cells and, of course, a number of CD8-positive T cells. And so with this in place, we moved to staining a tissue set which consisted of 12 breast cancer sections and 22 ovarian cancer sections. And I'm going to show a couple of examples of what we have seen. And so here's an initial example of a breast cancer section that's stained positive for all markers, where again we have PDL1 in green, which is shown right here, B7H4, which is co-expressed with cytokeratin, which is shown here. You can see the B7H4 in magenta, which is overlapping with the cytokeratin in cyan. And ultimately, VISTA is expressed in a series of sporadic cells, which is in an independent cell compartment compared to PDL1 and B7H4. And then there's a number of CD8-positive T cells within this environment as well. And so an important observation here is that PDL1, B7H4, and VISTA are all expressed in independent cell compartments, where again we have B7H4 co-expressed with the cytokeratin-positive cells. PDL1 is in a completely distinct set of cells, which from our previous and additional cell staining, typically is CD68-positive cells. And then VISTA is in a completely independent cell compartment as well. And as a note, this ultimately was an image composite, which is broken down ultimately when you're going through the image analysis into independent components, which are shown here. And so you are ultimately looking at each signal independently, where we have DAPI, PDL1, B7H4, cytokeratin, VISTA, and CD8 all broken down. And these come together to merge into this composite image. This is another example of a breast cancer section that we stained. In this particular case, this section was positive for B7H4 and VISTA, but negative for PDL1, so we were not detecting any of the green signal. Now moving into a couple of example ovarian cancer sections. Similar to the first breast cancer section that we looked at a couple of slides ago, this case is positive for PDL1, shown here in this little cell cluster staining green. Again, B7H4 in magenta, which is in cells that co-express cytokeratin. And then we have VISTA cells that are positive in a completely independent cell compartment from B7H4 and PDL1. And another ovarian cancer section shown here. Again, this one is PDL1 negative, but it's positive for B7H4, which is co-expressed again with cytokeratin. And then our VISTA cells are in a completely different cell compartment from our B7H4 cytokeratin positive cells. So those were a few examples. A quick summary of the staining is shown here, which is organized based on breast cancer and ovarian cancer, and which markers they are positive for, including VISTA in the left column, B7H4 in the middle, and PDL1 here on the right. And ultimately, within these panels, we found that in the breast cancer, 33% of the cases are positive for PDL1, 58% were positive for B7H4, and all had VISTA-positive cells. And interestingly, at least 75% of the cases were positive for 2 immune checkpoint control proteins. On the ovarian side, we saw 36% of the cases staining for PD-L1, whereas half of them were positive for B7H4, and then 95% of the cases were positive for VISTA. And again, 55% of the cases were positive for at least 2 immune checkpoint control proteins. And so we think this is a really important tool to look at the co-expression of the immune checkpoint control protein, because of course it would be very interesting to know which immune checkpoint control proteins are expressed in a particular patient's cancer. And if you have multiple immune checkpoint control proteins, this might be an important indicator that you would need to have a combination therapy approach. So, in summary, a multiplex IHC panel has been established to detect PD-L1, B7H4, and VISTA, along with CD8 and cytokeratin. And in our initial evaluation of breast and ovarian cancer, we saw co-expression of at least 2 immune checkpoint control proteins in 75% of breast cancer patients, or 55% of ovarian cancer patients. Importantly, PD-L1 and VISTA expression was observed in independent cell populations, while B7H4 is in yet another distinct cell compartment, which in this case is co-expressed with cytokeratin. And so we have a lot of follow-up panels in development, and so we're continuing to build our portfolio of immune checkpoint and immunotherapy relevant markers. Rabbit anti-mouse and mouse anti-human antibodies are a big focus as well. And we're also looking at alternative cell-based immunotherapy targets, like CAR-T targets. For example, our EGFR V3 antibody, an example shown here of staining in glioblastoma. So a couple of example follow-up panels. On the left here, looking at breast cancer, we have a panel looking at PD-L1 along with FOXP3 and CD8. There's a lot of interest in the ratio of effector cells to Treg cells, and so this panel is designed towards looking at PD-L1 in the context of the CD8 to FOXP3 ratio. And another panel here on the right, looking at PD-L1 along with CD3 and CD8. So CD3 and CD8 are very important basic immunology markers and comprise the immunoscore, and so it's looking at PD-L1 with these relevant markers as well. And finally, here's an example of human lung cancer that was stained with PD-1 and PD-L1, along with CD3, CD8, and cytokeratin, and then marking the nucleus with DAPI. And we can see this nice example up in the top right corner, which is the one up here, looking at PD-L1 in magenta, which is staining in a completely distinct compartment from the cytokeratin positive cells. And we can see a number of PD-1 positive cells in green, and then around this particular patch of cells. So at this point, we're going to ask one more poll question, and it's going to appear on your screens in a moment. And with that, I'd like to thank everyone for attending today and for your time and attention. And if you have any questions, I'd be very happy to field them. And my contact information is up on the screen at this point, which you can always follow up with me with questions as well. Thank you.
It was an excellent presentation, Dr. Silver. Thanks for bringing that information to us. Before we get started with all of your questions, here's a quick reminder of how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. Our first question comes from Jill Marinon from INSERM. Jill asks, are these performed with Tyramide amplification?
Yes. All of the experiments and images that I showed today were performed with the Tyramide signal amplification-based approach.
Great. Next we have Martin from NKI. Martin asks, IHC, even multiplexed as you have it, seems to have so many limitations. Of course, the spatial arrangement of cells can be very important and add valuable information, but the limitations on multiplexing due to bleaching or antibodies in the slide themselves. He says, I'm not very sure why there is such limitation on multiplexing and why you need to amplify the signal so much.
Yeah, the reason that we tend to feel that we need to amplify signal is due to autofluorescence that you'll see in a number of different tissues, or all tissues. Some, of course, are worse than others. Ideally, you'd be able to use directly conjugated antibodies, and this would simplify quite a number of things, and it would make quantitation more accurate. But the simple fact of the matter is, due to autofluorescence, you need amplification to rise above that noise. And so, again, the use of amplification is simply to provide you with a signal intensity to be able to detect your signal. We are, of course, interested in alternative approaches to basically step around the need for such extreme amplification. But at this point in time, Tyramide is a very nice solution to get that job done.
Okay. Next, we have Lisa from UVA who asks, are these tissues fixed with formalin?
Yes, they are fixed with formalin. The focus of the work that we've done and all the images that I've shown have been on formalin-fixed, paraffin-embedded tissues. So yes, they are fixed with formalin.
Great. Next we have Krishna from MPI Research, who asked if this webcast is going to be made available later. And I think the answer
yes, it is.
Yes there.
Yeah, the answer to that question is yes. This webcast will be made available.
Okay. Next, we have a question from one viewer who asked if there is a limitation, or is there a limit to the number of targets that can be detected? Are there any hardware or software restrictions to multiplexing?
Absolutely. The hardware is a very important factor that dictates the degree to which you can multiplex. As I mentioned, we're primarily using multispectral imaging, which enables us to look at 5+ colors. I think getting up to 8 colors at this point is possible. We don't quite have the algorithms in place to do this. If you're using standard imaging approaches, you can look at roughly 3 colors plus DAPI. But then you start to run into a lot of problems with the overlap of fluorochrome emission using a standard microscope. So yes, the hardware is an important factor that dictates the degree to which you can multiplex.
Great. One other viewer asked, how do you go about selecting a reliable antibody for mIHC? Are there antibodies against targets that are particularly difficult to work with?
Well, the first part of that question, the way that you go about it really is you need to identify high-quality, highly validated IHC antibodies. And so, while I'm probably a little biased, I think the best place to go is to CST for those antibodies. And really what it comes down to is you need to trust the validation work that has been done. And so if the antibody works really well on your standard IHC experiment, so a basic chromogenic IHC assay, we've found that these antibodies translate over very nicely to the TSA-based multiplexing approach. There's some more validation work that needs to be done. Often the optimal working concentrations change because the detection chemistries are different and the multiplexing workflow is slightly different. But ultimately, if you have a good IHC antibody, it will work well for this TSA-based multiplexing approach. And then to the second part of the question, are there any targets that tend to be particularly difficult? There are certain antibodies. I mean, certainly phospho-specific antibodies are challenging, but that's more of a problem for the development of the IHC antibody in the first place. Once you've cleared that hurdle and you have a validated IHC antibody, we haven't found any targets that haven't worked well in this approach.
Okay, great. There are no more questions. I would like once again to thank Dr. Matthew Silver for his presentation. Do you have any final comments for us?
I do not. I'd just like to thank everyone again for their time.
Great. I would like to thank our speaker today for today's informative presentation, as well as our sponsor, Cell Signaling Technology, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through July 2016. You will receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who aren't able to join today. Thanks for logging on and participating in today's broadcast event. See you next time.